Hi everyone, I'm Alston from Sino Biological. Today I'm going to talk about the chimeric antigen receptor associated cellular immunotherapy and how people use it as a formidable weapon to fight cancer. To cover this topic, I will give a brief overview of the CAR-associated immunotherapy and then talk about some CAR T and CAR NK therapy in clinic and then discuss the current limitations and future directions. Finally, I will introduce how Sino Biological is supporting the research in the CAR-associated cellular immunotherapy. Let's begin with the overview of CAR-associated immunotherapy. As we all know that CARs are designed to recognize specific antigens expressed on the cancer cells. CARs target tumor cells by binding to the receptor like CD19, HER2, EGFR, triggering tumor cells apoptosis through immune response. There are 4 different CAR structures, and each one of them have different functions to ensure the efficacy and specificity of the CAR therapy. According to this figure, we can see that the extracellular domain is usually derived from an antibody that specifically to a particular antigen expressed on the surface of cancer cells. And the hinge region contacts the extracellular antigen together with the transmembrane domain and provides flexibility to the CAR molecule, allowing it to move freely in the membrane. And the transmembrane domain is hydrophobic region that anchors the whole CAR molecule to the membrane of the modified immune cells, allowing it to interact with the intracellular signal machinery. So for the intracellular signal domain is typically derived from the signaling proteins that activate the whole immune cells upon recognition of the cancer cell through the extracellular domain and eventually initiate the signaling cascades that activate the immune cells to kill the cancer cells. The global CAR T therapy market size is expected to be $88.5 billion by 2032 and growing at around 30% annually from 2023 to 2032. As the marketing continues to expand, the CAR's engineering is evolving to enhance the specificity, efficacy, and potency of the whole immune therapy. Regarding to the CAR engineering, there are 4 generations of CARs. For the first generation CAR T cells, it consists of a single antigen recognition domain. Typically, this domain is from a single-chain antibody linked to a CD3 zeta signaling domain for T cell activation. While first-generation CAR T cells demonstrated some activity in the lab settings, but their clinical efficacy is limited due to insufficient signaling and antigen recognition. And in the second-generation CAR T cells, it improved upon the first generation by incorporating the co-stimulatory signaling domain such as CD28 or 41BB, in addition to the CD3 zeta domain. This design significantly enhances the CAR T cell proliferation and persistence, resulting in improved therapeutic outcomes observed in the clinical trials. And currently, all of the FDA-approved CAR T therapies are based on this type of construct. But for third-generation CAR T cells, further refine this design by integrating multiple co-stimulatory signaling domains into the CARs to further enhance the CAR T cells' activity and persistence. However, the advantages of third-generation CAR T cells are not as clear as the second generation. and the complexity of the production and clinical application increases. But for the fourth generation and beyond CAR T cells, they often referred to as the smart CAR T cells, optimized CAR structure and signaling mechanisms. These CAR T cells may possess self-regulatory functions, such as reducing activity in the presence of immunosuppressive agents or enhancing activity upon other stimulations. The evolution of CARs aims to improve the specificity, activity, and persistence of CAR T cells to achieve better therapeutic outcomes and broader the clinical applications. In the slides, I will briefly introduce the mechanism of multiple CAR-associated cellular immunotherapy, including CAR T cell therapy, CAR NK, and CAR M. Killing mechanism of these immunotherapy varies in different ways. For CAR T cells, activated CAR T cells can recognize the tumor-associated antigens expressed on the tumor cells, and cytotoxic activity of the CAR T cells is mediated by the GZMB and PFN, and by activation of the death receptor pathway, such as FAS or FASL, can further induce the tumor cells apoptosis and necrosis. But for the natural CAR NK cells, they are regulated by the signaling of activating and inhibitory receptors. For CAR NK cells, they also can secrete the PFN or GZMB, or through the death receptor pathway FAS trails to further enhance the cancer cell apoptosis and necrosis. They can also secrete the interferon gamma and TNF-alpha, similar as what CAR T cell, further enhance the efficacy of the whole therapy. And CAR M is another CAR-associated immunotherapy where the macrophage involved. The binding of a specific tumor-associated antigens with CAR receptor on the surface of the CAR M, it can generate the activation signal that mediate tumor phagocytosis. Activation of the transcriptional factor, the NF-kappa B, and subsequent release of pro-inflammatory cytokines, which in turn can activate the T-cell mediated immune response against the tumor. Unlike well-established CAR T production, CAR NK is still a newly approach and needs a lot of optimization. However, theoretically, CAR NK has a lot of advantages over CAR T, which make it promising alternative cell therapy compared to CAR T therapy. CAR NK are relatively safer than the CAR T therapy, since the conventional T cells require HLA matching to recognize the target cells, GVHD, which is graft versus host disease, is one of the prevalent toxicity of the CAR T cell therapy. Since CAR NK cells act independently of the HLA matching to recognize the target cells, and GVHD has rarely been reported in clinic and preclinical studies of the CAR-modified NK therapy. Another advantages of CAR NK is off-the-shelf effect compared to the CAR T. NK cells can be obtained from a variety of sources, including cord blood, peripheral blood, and other cell lines, which allows for the development of off-the-shelf CAR NK cell products. And it also does not need to obtain the NK cells from the patient. This further eliminates the need for individualized cell manufacturing and reduce the overall cost of the production. Actually, it was mentioned that the lower cost regarding the cell source the starting cell source for the CAR T cells, which are much less abundant in peripheral blood and require more extensive processing to isolate and expand from the patient. But for the CAR-NK therapy, which are already present in the peripheral blood and can be easily isolated and expanded. As the market continues to expand, novel therapy related to the CARs are constantly emerging and being validated in the clinical trials. There are more than 200 clinical trials being developed each year, targeting both hematological malignancies and solid tumors. With the CAR-T and CAR-NK in human cancers being discussed, please let me introduce more clinical performance of types of CAR-associated immunotherapies. In the distribution of cell therapy pipelines under developing, we can see that the CAR-T pipeline is significantly leading the clinic compared to all the other immunotherapies. Since the FDA approved the first CAR T-cell therapy, Chimeric, in 2017, there are currently 6 CAR T drugs approved by the FDA, targeting CD19 and BCMA. This innovative cell therapy brought a huge breakthrough in the treatment of hematological malignancies. Thus, it has led to great interest in the R&D for cancer treatment from the research and the whole industry community. CAR T-cell therapy has been rapidly developing in recent years. Unlike traditional cancer therapy, CAR T-cell therapy is considered as the living drug and personalized therapy, which uses the patient's own immune T cells to attack cancer cells. The entire process of the CAR T therapy can be divided into these 5 steps. In the first step, the T cells are collected and isolated from the peripheral blood of the patient or other allogenic donors. Using the genetic engineering techniques, the T cells are transduced and equipped with a GPS navigation system called a CAR, transforming the T cells into the CAR T cells. The CAR T cells are cultured and further expand in vitro to obtain an adequate numbers of cells for infusion. After the quality control testing of the CAR T cells, the prepared high-quality CAR T cells are infused into patients to targeting the cancer cells while avoiding damage to normal tissue. There are several success-determining factors regarding the CAR T-cell therapy development, including selection of antigen and antigen binding domain, cytokines enhancing the cell's activations and expansion, and eventually the CAR detection in the last step, quality control. Similar to CAR T development, the basic of CAR-NK manufacturing processing includes CAR-NK cells extraction and CAR structure design and validation, and transfection of CAR molecule into the NK cells, CAR-NK cells expansion, and eventually injection back to the patient after quality control. For every steps in the process could be a challenging part and determine whether a CAR-NK product can be successful. And I will address 3 of them today, which could be the most important factors during the whole process, also as I mentioned in previous slide. The selection of an appropriate antigens is a crucial step in the development of CAR-T or CAR-NK cell therapy. To ensure the specificity, the target antigen should be expressed exclusive or predominantly on the surface of the tumor cells, and not on the normal tissue, to minimize the off-target cytotoxicity. The target antigen should not be essential for normal cellular function to avoid potential toxicity to normal cells. In this table, there are some typical targets antigens which are highly expressed on the tumor cells. After the selection of appropriate antigen, how to develop the corresponding scFv is also very important. It requires high specificity and affinity to ensure efficient recognition of the cancer cells. To this end, the binding affinity needed to be tested before the scFv can be constructed into the CAR molecule. Here shows an example of the CAR-T development of EGFR variant 3. So researchers used SPR to determine the affinity between scFv and EGFR and their target EGFR variant 3. So SPR shows great binding of the GCTO2 scFv to the recombinant EGFR variant 3. But compared to the negative GFP control and no binding to the EGFR wild type protein at the concentrate tested. It turns out that the scFv only bind to the EGFR variant 3 with good affinity, but not EGFR. This validation is very useful in the CAR development. After the whole infusion of the CAR T cells to the mice, the tumor size decreased a lot. Since the NK cells represent only a minor fraction of the peripheral blood, thus the generation of sufficient number of NK cells remains a major challenge for the adoptive immunotherapy in humans. Currently, despite the various methods available to promote NK cell growth, the most widely used approaches is cytokine mixture induction. On the left side, we can see that the researcher has investigated on the function of the IL-2 and IL-12 during the proliferation of the NK92 cells. The results show that after adding the IL-2, the cells show dramatically increase of the expansion or the proliferation. And a similar effect can also be shown in the group with the IL-12 treatment. Besides promoting cell proliferation, cytokines can also increase the NK cell cytotoxic by promoting the activation. On the right side, we show an example. The researcher added a mixture of the cytokines IL-15, 18, and 27 to stimulate the NK cell activation. The expression level of the perforin and granzyme B also dramatically increased or enhanced regarding this treatment of the mixture of the cytokines. The next important part during the CAR therapy production is CAR detection. This step is to assess the expression of the CAR construct, which is a very important step in quality control. CAR expression level and consistency in CAR-T and CAR-NK cell therapy needed to be carefully monitored to ensure that the cells will function as intended in the clinical setting. Detection of the CAR expression provides an important quality control measures to ensure that the cells meet the required specification before or after they used for the treatment. As I mentioned, detection of the CAR expression on CAR-T or CAR-NK cells after infusion is also needed. This can provide the information about the persistence and distribution of the cells in the body. This information can also help in monitor the therapeutic efficacy of the therapy, as well as identifying potential reasons for the treatment failure. There are several methods we can use to monitor the CAR expression, including the flow cytometry, which is a very convenient and widely used in the CAR development. And we can also use the Western blot, qPCR, also the confocal microscope are also like the common used technique or applications we need to further validate. We have briefly covered how CAR-T or CAR-NK therapy being developed. Let's see what kind of challenges are CAR-associated immunotherapy facing overall. CAR-T cell therapy related challenges include cytokine release syndromes, tumor-associated antigen escape, trafficking and tumor infiltration, on-target off-tumor effect, and immunosuppressive tumor microenvironment. And cytokine release syndrome is associated with the choice of the co-stimulatory domain as well as the length of the hinge domain. To overcome this, cytokines released by the macrophages during the pyroptosis can be mitigated by using specific drugs to target different cytokines, as I listed here. As we know that CAR-T cells mediated targets cell apoptosis when target antigen is present on the surface. Tumor antigen escape in absence of the surface antigen, and potential strategy to overcome this is to use the dual CARs or the bicars. Reduced homing of CAR-T cells to the tumor microenvironment due to the presence of different cellular components, including MDSC or Treg cells, can be observed in the clinic. To improve homing of the CAR-T cells to tumor microenvironment, we can consider using armored anti-tumor, anti-angiogenic CARs, as well as self-driven or self-driving CARs, which express multiple anti-angiogenic factors. But for the on-target, off-target effect, this might be mitigated by selection of reliable tumor-associated antigens by integration of the AI or artificial intelligence with big data mining. For the suppressive tumor microenvironment, it comprising different cellular components, including low oxygen, cancer-associated fibroblast, high ROS, or other components that diminish the proliferation of the CAR-T cells. To overcome this, CAR-T expressing anti-checkpoint inhibitors to promote the growth of the T cells in the tumor microenvironment, or the HIF-1 alpha inducible CARs, which can also get activated in the hypoxic tumor microenvironment, and the catalyst expressing CARs to scavenge the ROS in the tumors to promote T cells growth can all be utilized in the practice. To alleviate the effect of the cytokines release syndrome, the glucocorticoids or cytokine-targeted approaches are used. In vitro and in vivo studies have suggested novel cytokines-targeted approaches for the mitigation of the CRS. Those are directed against the cytokines including IL-1 beta, TNF alpha, interferon gamma, GM-CSF, by lots of small molecule inhibitors. Along these lines, the TNF alpha and the interferon gamma and GM-CSF knockout can be engineered in the CAR-T cells to prevent the CRS as well. The dasatinib is an FDA-approved tyrosine kinase inhibitor, has been investigated to inhibit the phosphorylation of immune receptor tyrosine-based activation motifs, eventually reversibly inhibiting the CAR-T cells to avoid CRS. According to the figure A, the dasatinib treatment temporarily inhibit the cytokine production. More importantly, upon the suspension of dasatinib administration in vivo, CAR-T cells effectively recover their functionality in tumor suppression. To overcome the tumor-associated antigen escape, CARs are engineered to recognize two different antigens simultaneously using bispecific dual or tandem CAR-T cells. By targeting multiple antigens, the likelihood of the tumor cells escaping recognition decreases. One of the most promising application of the dual CARs is using the dual CD19 and CD123 targeting the B-cell acute lymphoblastic leukemia. Firstly, both CAR-T19 in green and CAR-T123 in red were engaged in the same immunosynapse, merged as yellow in a dual CAR, as shown in Figure A. More excitingly, when tested in the in vivo, the dual-stimulated CAR-T19-123 cells exhibit higher efficacy against the BALL in the comparison with an equivalent number of the single-stimulated CAR-T19 or CAR-T123 or even the pooled CAR-T19 plus CAR-T123 therapy treatment in the cells. In order to increase the tumor specificity and reduce the off-tumor toxicity, the synthetic notch or so-called SynNotch CAR being produced and applied. In this figure, encounter of the primary antigen marked in purple leads to the translocation of the transcriptional factor into the nucleus, which induced another CAR expression on the surface to recognize the second signaling from the tumor. And the T cell activation and tumor cells killing only occurs in the presence of both antigens, which spars healthy tissue that only express the primary antigen. The SynNotch CAR-T therapy has been successfully applied on glioblastoma. Researchers implanted the GBM6 tumor into the brain of the NCG mice, treated them with the T cells bearing the EGFR-V3 SynNotch and the tandem CAR circuit. As controls, they treat the mice with the non-transduced T cells, or T cells constitutively expressing either EGFR-V3 CARs or tandem CAR. Treatment with the constitutive FA2 IL-13 alpha-2 tandem CAR was largely ineffective. However, treatment for the constitutive EGFR-V3 CAR T cells only show initial tumor shrinkage, but consistently results in a recurrence. But in contrast, all of the mice treated with the SynNotch CAR T cells showed complete remission of the GBM6 tumors. On-target off-tumor toxicity has been widely discussed in the CAR therapy, as I mentioned. One of the possible solutions is to target the intracellular antigens by using the genetic engineered TCR T cells. Given that TCR targets the intracellular peptides expressed on the tumor MHC molecules, this provides an expanded pool of potential targetable tumor-specific antigens related to the cell surface antigens that are targeted by the CAR T cells. T cells' cytotoxic activity depends on the TCR affinity. Different methods have been developed to optimize the TCR affinity. TCR optimization can be performed by modifying the constant region of the both TCR alpha and beta chains to prevent mispairing and even enhance the TCR expression and stability. Most recently, mutations in the variable regions but outside of the CDR region have been shown to improve the TCR expression and stabilities. Once the TCR have been identified, in vitro and in vivo functional assays validate the specificity and the efficacy of the engineered T cells. There are a lot of promising target antigens under the scope and being validated via multiple clinical trials. Such target can also be used to treat many solid tumors. Another promising approach to enhance the CAR-T efficacy is combination treatment with other therapeutics, such as chemotherapy, oncolytic viruses, or radiotherapy, or even immune checkpoint inhibitors. Chemotherapy is known to increase the expression of the tumor-associated antigen on the cancer cells. This effect will help the enhancing the CAR T cells interaction with the cancer cells and promoting the CAR T cells proliferation, infiltration, and extend their persistence in the tumor microenvironment. And through the oncolytic viruses, the tumor debulking further enhance the CAR T cells infiltration, proliferation, and activation. The cytokines production and cancer cell apoptosis. The radioactive therapy induced the chemokines, interferon gamma, DAMPs released by the tumor cells, leading to the increased migration and infiltration of the CAR T cells. Eventually, the inhibitor of the immune checkpoint targeting the interaction between PD-L1, PD-1 alleviate the inhibition of the CAR T cells. This effect enhance the CAR T cells activity and consequently promote the cancer cell death. Similar to what we discussed in previous slides, a combination of CAR NK cells with other therapy also show promising outcomes. For example, with the chemotherapy, the upregulation of the tumor-associated antigens on the cancer cells further enhance the CAR NK cells' interaction with the cancer cells and promote their cytotoxic activity. Moreover, by decreasing the Treg and MDSCs, chemotherapy further promotes the CAR NK cells' proliferation, infiltration, and prolong their survival in tumor microenvironment. Oncolytic viruses also being mentioned in the combination approaches that tumor debulking induced by the oncolytic viruses further enhance the CAR NK cells infiltration. Now I would like to take this opportunity to highlight how Sino Biological, as a leading reagent company, is supporting the CAR-associated immunotherapy. Sino Biological is dedicated to providing one-stop research support, and now we have covered almost all the reagent and services needed during the CAR T or CAR NK production, through CAR development all the way to CAR T CAR NK cell quality control. Especially we perform for antibody development platforms and more than 270 HPLC verified target antigens with high bioactivity to facilitate the CAR development. We also offer GMP grade supernuclease and ELISA kit to support lentivirus packaging. To support the cell activation and quality control, we also offer GMP grade cytokines, FACS antibodies, protein L and anti-ideal type antibodies through our catalog products or other CRO services. As we know, during the CAR T and CAR NK production, there are several key factors that determine the success of CAR T and CAR NK products. Sino Biological is providing products and services to help our customer to overcome all the challenges and get the best result. To facilitate the discovery of the SCFV, we have 4 platforms as I mentioned, including phage display, Beacon B cell system, and FACS B cell screening, and hybridoma platform. Since the SCFV sequence of the most of CAR T or CAR NK cells are derived from mice and have certain immunogenicity, SCFV needs to be further humanized. Since, as I mentioned here, Sino Biological provide high quality monoclonal antibody humanization services that enable a high degree of successful humanization. As we shown before, a crucial determination of CAR functionality is the affinity of the SCFV to its target antigen. Sino Biological also provide the SPR or bleed assay services which can effectively screen appropriate CFB molecules. To facilitate the T cells or NK cells expansion, Sino Biological also provides GMP-grade cytokines. Since cytokines needed to be added into the cell culture and cell product would be infused back to patient, the quality of the cytokines should be strictly controlled. Now we have 4 systems, including GMP-grade quality management system, quality control system, and the product safety testing system to further ensure the high quality, purity, safety cytokines. To facilitate the CAR detection, Sino Biological now has almost all the CAR-T CAR-NK related target protein with high purity and labeled with biotin, which is ready to use to detect the CAR molecules on cells. Besides the variety of the target proteins, we also provide customized services to develop anti-ideotype antibodies, which can recognize the CAR molecules in no time. ELISA kits are used to measure the amount of cytokines such as TNF-alpha, IL-2, interferon gamma, and other cytokines provides insight into the specific immune response. Sino Biological offers a variety of validated ELISA kits and antibody pair sets developed against a lot of cytokines to monitor and detect the residual cytokines. The evaluation of the CAR-T and CAR-NK cells cytotoxicity involves 2 main methods: cytokine release detection and in vitro tumor cell killing assays. The first method evaluates CAR-T expressed cell activation and their ability to produce cytokines such as interferon gamma, TNF-alpha, while later measures the ability of the CAR-T or CAR-NK cells to kill tumor cells by incubating them together with and qualifying the tumor cell deaths. To support the evaluation of the CAR-T, CAR-NK cell therapy cytotoxicity, Sino Biological offered high-quality cytokine ELISA kit, as well as the cell line development services for cell killing assay development. An extensive catalog. We offer over 7,000 proteins, 14,700 antibodies, more than 47,000 genes, and another 600 ELISA kits, providing a diverse selection of the tools to support your research needs. Our vast collection covers various species, including human, mouse, rat, and more, which allowing you to explore different biological systems and pathways. From recombinant protein to monoclonal antibodies, our high-quality products undergo rigorous testing and validation to ensure the reliability and consistency. With our extensive portfolio, we aim to provide researchers with the necessary tools to advance scientist discovery and drive the innovation in the field. Our CRO services are designed to support your research and development needs across various fields, including the CAR therapy development. We offer a range of services, including the recombinant protein expression, recombinant antibody production, monoclonal antibody development, and polyclonal antibody development. With our expertise and state-of-art facilities, we provide reliable and efficient solutions to accelerate your research projects. As a global company, our research extended to over 90 countries, and we have established a strong presence worldwide. We are committed to delivering high-quality products and services that meet the needs of the researchers and scientists around the globe. We are proud to see our products cited over 14,000 publications appearing in high-impact journals. This recognition demonstrates the trust and the confidence researchers have placed in our reagent and services. Our commitment to quality is evidenced in our ISO certifications. These certifications reflect our dedication to maintaining rigorous quality management systems to ensure the consistency and reliability of our product and services. At Sino Biological, we have established a strong global presence, with our headquarters located in Beijing. In addition to our main office, we have placed branches in various cities to better serve our customers. Our branches can be found internationally in Japan, Tokyo, Frankfurt, Germany, and in Pennsylvania and Texas in the United States. This location enables us to provide efficient and reliable support to our customer worldwide, ensuring proper delivery of our high-quality bioreagents and comprehensive CRO services. With our global network, we are dedicated to facilitate scientific advancements and collaborations across the board. We would like to express our sincere gratitude for your time and attention today. If you have any further questions, require additional information, or would like to explore our offerings in more details, please don't hesitate to reach out to our team. We are here to assist you and provide tailored solutions to meet your research needs. Thank you once again for your participation and interest. Thank you.